Structure and functions of neurons. Structure. Whatever the type, neurons share the same basic components. They have a cell body, or soma, which holds the nucleus containing genetic material, and protruding from this are extensions that act to transmit the electrical signal that is the function of the neuron. In one direction are dendrites. These branch-like structures carry information from organs and other neurons towards the cell body. The other extension is called the axon. This acts to carry the nerve signal or action potential away from the cell body, either to another neuron or muscles or organs. At the end of the axon is a terminal button or synaptic knob. This is the part which forms a synapse with the next nerve or other body tissue. In the case of skeletal muscle, this is a different shape and is called a motor end plate. One last thing to comment on at this stage is the branch-like projections of dendrites and axons can be insulated with a fatty tissue sheath called the myelin sheath. This acts to speed up transmission of the action potential, the electrical signal. But in order for this to work, it's important that there are gaps in it, called the nodes of Ranvier. We will look at these later. Nerve cells in the CNS, the central nervous system, are less likely to have myelin than those in the peripheral nervous system. Can you think why? And it's not to do with the speed of transition. Have a think what it would look like looking at an insulated pipe next to an uninsulated pipe. That might help. You need to understand the structure and function of three different types of neuron: sensory, relay or interneurons, and motor neurons. Let's imagine a bit of old school breakdancing. These three characters are going to represent our neurons for now, and they want to send a wave down their arms. The wave is going to travel in one direction only, and this represents the action potential travelling between the three types of neuron. Let's imagine nerve cell 1 touches something hot, and this triggers the wave. Nerve cell 2 is going to pass the signal on to nerve cell 3, and there will be some result at the end. So nerve 1 is the sensory nerve. Nerve 2 is the relay. It relays the input between the other two. And nerve 3 is the motor neuron. Okay, let's get a little bit more technical. The sensory neuron carries sensory information from the environment or organs to the central nervous system. They can be distinguished because they have long dendrites and short axons. The relay neuron connects sensory and motor neurons, usually within the CNS, and have a short dendrite and axon. The motor neuron carries instructional signals from the central nervous system to skeletal muscle and internal organs and glands. They have short dendrites and long axons. Why do you think it might be arranged like this with the cell bodies where they are? Look where all the cell bodies end up being, near the middle towards the spinal cord. Why do you think that might be? Electrical transmission along a nerve. This is essentially a wave of changing polarisation travelling along the nerve as the balance of ions across the nerve membrane on an axon or dendrite changes. In its resting state, the inside of the axon or dendrite is slightly negative compared to the outside. The beginning of a nerve signal, we'll look at this later, causes a point on the nerve to become open to sodium ions, which cross into the neuron, making the inside slightly positive. This depolarization triggers the same process in the next bit of the neuron. So the wave of positive charge flows along the nerve. A similar wave of repolarization follows rapidly on behind this to return the nerve to its original state and ready for the next signal. In nerves with myelin sheaths, this wave of depolarization jumps from gap to gap, as these are the points where the ions can cross the membrane. This has the effect of speeding up nerve transmission. When the action potential gets to the end of an axon, we can then look at how a signal crosses from neuron to neuron. These junctions between nerves or nerves and tissues are synapses, and you should have some familiarity with these from GCSE biology. The synapse refers to the junction. We have an action potential travelling down the presynaptic nerve, releasing neurotransmitters into the synaptic cleft, which then attach to receptor sites on the postsynaptic neuron, where they will have an effect on the activity of that neuron. Let's look at this in a little bit more detail. The action potential causes a synaptic vesicle which contains neurotransmitters to fuse to the presynaptic membrane of the terminal button and release the neurotransmitter into the synaptic cleft. Receptor sites on the dendrite of the postsynaptic neuron are specific to different neurotransmitters, like a molecular lock and key. Specific neurotransmitters will have a specific effect, either increasing or decreasing the chances of action potentials in the postsynaptic neuron. Though we tend to learn this in a simplistic way, the actual effects for a particular neurotransmitter can be different in different parts of the brain. Many drugs have their effect by either mimicking a neurotransmitter or influencing synaptic transmission in some other way, and we'll look at some of these examples later. First, let's look at these specific effects a neurotransmitter can have on the postsynaptic neuron. If the neurotransmitter reduces the chance of an action potential in the postsynaptic neuron, then we call this an inhibitory neurotransmitter. In fact, much of the activity in your brain is inhibitory, with some bits telling other bits constantly to shut up. If the neurotransmitter increases the chance of an action potential in the postsynaptic neuron, then we call this an excitatory neurotransmitter. This is, of course, where the process of depolarization of the nerve membrane for the next action potential starts. However, in reality, we don't really have a nervous system made up of one nerve talking to another nerve like we see in nice textbook diagrams. And a single postsynaptic neuron could have many excitatory and inhibitory neurons all trying to send instructions at that particular synapse. The decision of whether there is a postsynaptic action potential is based on a process of summation, which is a calculation of all the excitatory and inhibitory influences at a particular moment. As a typical example, we can consider dopamine as an example of an excitatory transmitter that is linked to several functions including motor behaviour, pleasure, motivation and emotional arousal. Low levels have been linked to Parkinson's disease and higher levels have been linked to schizophrenia. Serotonin causes a postsynaptic neuron to become more negatively charged, and it is therefore less likely to generate an action potential. It's therefore an inhibitory neurotransmitter. It's connected with a very wide range of activities, including the regulation of sleep and appetite, memory and learning, mood and emotional behaviour. Low levels of serotonin have been suggested to have a role in depression. Let's finish by looking at some drug actions at synapses. For example, antipsychotic medications. Many of these work by blocking dopamine receptor sites. They occupy the receptor sites but do not trigger a postsynaptic action potential. The fact that these drugs have been effective in reducing certain symptoms of conditions like schizophrenia has led to the idea of the dopamine hypothesis, that conditions like schizophrenia are caused by high levels of dopamine activity. Another very interesting drug is curare. This is a powerful neurotoxin of plant origin. It works by mimicking acetylcholine at the junction between motor nerves and muscle fibres. It sits in the acetylcholine receptor sites, but it doesn't again trigger a postsynaptic action potential. This means that the muscles can't contract, and the victim will die of asphyxiation or suffocation. There is a practical application to the use of this drug in that it can be used in surgical procedures to stop muscles contracting. It's a muscle relaxant. Finally, we could consider selective serotonin reuptake inhibitors, or SSRIs, like Prozac. Reuptake is a mechanism by which a presynaptic neuron can make sure a signal is transmitted, but there isn't an excess of neurotransmitter left in the synaptic cleft. SSRIs have their effect by blocking these reuptake sites and therefore increasing the amount of available serotonin at the synapse. The fact that this might be effective in reducing the symptoms of depression may imply that depression is caused normally by low levels of serotonin.